Welcome everybody to another Kansas State Veterinary Diagnostic Laboratory webinar. I'm Greg Hanselcheck. I'm the moderator today. For those of you that have been with us in the past, appreciate you taking another time out of your busy schedule to be with us. For those of you that are new to our webinars, just a little bit of bookkeeping things. One is that we try to put a new webinar up or have one every other month or as needed with timely topics like today's topic. These videos are recorded. If you stay with us the entire webinar, you will get an hour of CE credit, and we will send you a certificate in 3 to 4 weeks. The recordings will be placed on the KSU CVM Continuing Education website, and if you watch the webinar there and then take a short quiz, you'll also get some CE. These videos will also be on the KSVDL YouTube channel. Unfortunately, if you watch it there, you won't get credit for the CE part of the program. But so some other things is that if questions during the period, this is set up so that we want the questions at the end of the presentation. So during the presentation, if you have questions, just go to the top of your screen there and enter them in the Q&A. That'll be anonymous. If you want to do the chat, that's fine. It's just that you'll be identified on who's asking the question. The other part of this is that at the end of this, you will be asked to complete a survey, a very short survey. And the reason we're doing that is that we want to know what we're doing right and what we can improve on. But probably the most important part of that is that please give us topics that you would like to see the diagnostic lab at K-State provide for you in the future. So now it's my great pleasure to introduce our speaker for today, Dr. Mike Apley. For anybody, whether you're a producer or a veterinarian that's been around production animals for more than about 2 minutes, you know who Dr. Apley is. Mike is the head of our anatomy and physiology department now here at K-State. Mike got his DVM here and his PhD in pharmacology. He's been a practitioner. He's been a researcher. He does a lot of teaching in a lot of our production animal medicine courses. He's also the coordinator of our advanced feedlot course here at K-State. He's also been the president of the ABP, president of the AVC, president of the President's Council on Antimicrobial Resistance a few years ago. And then in December of last year, he was actually awarded the AVC Production Animal Consultant of the Year. So he's got, and he spends a lot of time talking to the FDA and the USDA and the people in regulation. So the fact we received the Dear Veterinarian letter about aspirin, we thought he'd be the perfect person to come in and explain why this has happened and maybe what alternatives that we have as food animal veterinarians to use analgesics. So with that, I'll turn it over to Dr. Apley.
Thanks, Dr. Hanselcheck. Well, as Greg said, the goal today is to talk about what's gone on with aspirin and our other alternatives, go through some regulations with that. But start with sorting back through what happened. And this letter that came out, I think our veterinarians online here would recognize that. And the FDA communicated with veterinarians saying they've become aware that veterinarians and dairy farmers are treating lactating dairy cattle for pyrexia and pain with aspirin, and they wanted to clarify that there are no FDA-approved aspirin products for use in cattle. And this is the key sentence there that made it clear that this applied to more than just this instance in cattle, and that's that the extra-label use of unapproved drugs in food-producing species is prohibited. They point out that there are FDA-approved products for controlling pyrexia and pain in lactating dairy cattle, safe, effective, established milk and meat withdrawal periods. And as veterinarians, we're well aware that one of our main objectives, our main obligations in working with food animals is to make sure that meat, milk, or eggs derived from those food animals are safe for human consumption. And one of the ways we do that is by making sure that the withdrawal period has been established and is enforced for those animals. And in the case of extra label use, we are required to work to find those data that would allow us to create that exaggerated withdrawal time. So you can see there's just the way you would say if it is actually approved. So for animals, it's a new animal drug application number that's approved, or an amended new animal drug application for a generic. And for humans, it's NDA, just without the animal. It's the same deal that tells us it's an approved animal drug. They go on to point out, reference the Animal Medicinal Drug Use Clarification Act, or AMDUCA, and that we are allowed to use drugs off-label, but they have to be an FDA-approved human or animal drug. They have to have that NADA or NDA or the amended ones that represent their generic. There is one FDA-approved human aspirin product, but that is one that would not be practical to use in cattle or food animals. The rest of them are marketed under an OTC monograph, which we'll talk about in a minute. So it's also, they talk about the impracticality of dosing cattle with a sufficient amount of the approved human product. What can be used, or what was used, were the bulk pails of aspirin, which could then be kept in a suspension. Acetyl salicylic acid is not water soluble, sodium salicylate is. So the acetyl salicylic acid you would have to keep agitated and in suspension to use. So the FDA looked at this, and it previously had been granting some regulatory discretion. And the way I had it explained to me is, if you're driving 63 in a 55 and you pass an officer of the law and they choose not to enforce the 55, they're not telling you that 55 is, or 63 is legal. It is a decision on enforcement. And in this case, seeing that their information was it was being used in these cattle on a widespread basis, they elected to come out and say that there will not be regulatory discretion, and that this is an illegal drug to use, since it has no FDA approval for the specific products being used. And that's how that came about. It was becoming aware of the use that was going on, and also the publicity surrounding that use. And they elected to come out and say, well, aspirin is not approved, so therefore under AMDUCA, it's always, as it always has been, that use of this drug in an extra label manner is not permitted under the AMDUCA regulations. So they said you should stop the use of unapproved aspirin in lactating dairy cattle and use FDA-approved products to control pyrexia and pain. And of course, this was associated with the influenza that was going through the cattle. And if they've already been treated, you were supposed to get your extended withdrawal time in order and apply that to the dairy cattle themselves and the milk. So it came about from a turn in how the agency was perceiving this use, and electing to say, now this is how we will enforce and go forward the fact that you cannot use this in an extra label manner. So there's a few things I wanted to cover as we go back towards this, talking about where this came from and how it's affecting us. You know, really, aspirin has not been one of our primary anti-inflammatories for use in cattle. This was a special situation where they were looking at large numbers and how it might be applied, but it's not our best NSAID for cattle. We'll go through why. But I just wanted to cover for a minute all the animals that are considered food animal species. And this is a guidance document from the FDA. And in that, they go through all the ones that they consider food animal species that are minor species, and that includes bison, deer, elk. Of course, goats and sheep are minor species. You're all well aware of that. Rabbits, a lot of birds are game birds, alligators, turtles, frogs, honeybees, sea urchins, and sea cucumbers. So it's a interesting list of the things they consider to be food animals. And they note at the bottom that this is not an exhaustive list, but ones that they currently consider, and others might be. So AMDUCA applies to food animals. There are some things where minor species are split off in some prohibitions, and of course in minor food animal species we're able to use drugs in the feed off-label under Guidance for Industry 615.115. But as far as Amduca goes, and what we're going to talk about today with aspirin, that applies to all of them. And so just as a reminder how Amduca works for us, and I'm talking to a group of people that are experts in it, but just to clarify how it applies to this, is as you start out, these are from Part 530, that they specifically address things that would apply here. And they say that permitted extra-label animal and or drug use in food animals is okay when there's no approved new animal drug that is labeled for such use and that contains the same active ingredient which is in the required dosage form and concentration, except where a veterinarian finds within the context of a VCPR that the approved new animal drug is clinically ineffective for its intended use. And prior to prescribing, there's a list of things there you need to do. The second one is establish a substantially extended withdrawal period. One of the things that I think we should be clear on when we look at the part where it says where a veterinarian finds within the VCPR that the approved new animal drug is clinically ineffective for its intended use, that is a decision by the veterinarian within their scope of professional practice. So the FDA regulates distribution of drugs, they regulate drug approval, they do not regulate the practice of veterinary medicine. And your decision on whether that labeled drug is effective or not is the practice of veterinary medicine. So I always encourage veterinarians that you should be confident in that your decision there is not an FDA regulated decision. How you go down through the drugs, whether or not it's approved for food animals or not, those are FDA regulated, but that decision of whether or not that label one is effective is yours as a veterinarian within the practice of veterinary medicine. Then there's other conditions, and for example, if you can't come up with withdrawal time information, you can't use it, obviously. And here's the big point that I think applies here, that extra label use of an approved human drug in a food-producing animal is not permitted under this part if an animal drug approved for use in food-producing animals can be used in an extra label manner for the particular use. So Amduca has 4 steps. One is use the labeled drug for the labeled application. Everything fits on label. Number 2 is if that labeled drug is ineffective as determined by you, the practicing veterinarian, then you're expected to first consider the use of another food animal drug, okay, in an extra label manner. And then once you get past that and you say there's no other food animal drug that I can use in an off-label manner, then you can use equally either a non-food animal veterinary drug or a human approved drug. The purpose there is that we would use a drug first that has gone through some type of food animal approval so that there is a withdrawal time in at least one species, and there's been toxicity work, human withdrawal time considerations done in at least one species. So that's one of the big points of Amduca that I think applies here. And compounding. Some of the uses of aspirin could be considered compounding, and that's the 4th step of Amduca. So you said there's no label drug I can use, there's no food animal label drug I can use in an off-label manner. I can't use a non-food animal veterinary or a human drug in an off-label manner as prepared. Now I have to compound. And when you compound, it's similar in that when you compound, you're expected to compound from a food animal, approved animal drug first, an approved animal drug before you use an approved human drug. But the big thing to make a point on here is what ended up being the problem for aspirin is coming back and saying, yes, it is not approved. The products that are being used are not approved for food animals, and they're not approved anywhere, right? The aspirin that you get in a jar is not an approved product. The aspirin that you could pick up at the farm store never was approved by the FDA. So yes, it was sold, but it was not approved. And that is where some of the confusion's been. So when we look at the salicylic acid derivatives, they were the first NSAIDs used in modern medicine. And again, veterinary forms have never been formally approved by the FDA CVM. Common dose rates for 50 to 100 milligrams of aspirin per kilogram. And one of the things I wanted to address is, before we get too wrapped around the axle, actually looking at how effective it is, right? So we go look at this 50 mg per kilogram Q12H, which would be a pretty low dose. That's one 240 grain bolus per 686 pounds. I think it would be a lot less than people have used in the past with those. But if you look at our expected concentration, which would give us some relief, at least from arthritic pain, other pain type syndromes, and so that gray area would need to be above that gray area to have that effect. And you fill in how often that is, and you're looking at brief periods of 2 to 3 hours after it's administered, where we would actually think that we had some type of effect. And dose accumulation is pretty quick for a drug with about a 3-hour T1/2 in the plasma once absorbed. So you look at 7 T1/2s to almost 100% of steady state. You're to steady state within a day going orally. So that's that dose. Here is 100 mg per kg, which would be one 240 grain bolus per 300 pounds. I know in the past people have done like 1 per 200. So if you go at this dose, now you've got a lot higher concentrations being achieved, and the higher you get, the longer you can ride out over any given concentration. And you're looking at being out there, maybe 7 or 8 hours after a dose. So at this dose, you'd need to give it at least 3 times a day to maintain the concentrations you wanted. If you went 1 per 200 pounds, you'd probably be where you could go twice a day and be above that 30 micrograms per mil fairly effectively. But it would take a bolus per 200 pounds going Q12H to make that work, that to happen. So frequent administration with some fairly high doses would need to happen. One of the points of confusion is grass and gray, so generally regarded as safe and generally regarded as effective. So in veterinary medicine, grass and gray primarily applies to feed additives, compounds that are added to the feed that are generally regarded as safe and generally regarded as effective. And aspirin is neither one of those. But when you get into the human side, there's grassy, but generally regarded as safe and effective. And there's 2 regulatory pathways to bring a non-prescription drug to market in the U.S. So this is non-prescription drugs. And if you go to that website, if you just Google over-the-counter monograph, you'll come to the pages I'm going to show you. But there's a drug application process, so the new drug application or the new animal drug application, and there's the over-the-counter OTC review monograph process. And these are for the human drugs that are over-the-counter. So for example, you go buy a topical cream for a scratch you have that has antibiotics in it, that's marketed under an OTC monograph process, since it's not prescription. Your cough medications, drugs like that, are OTC monographs. So there's this is the page you go to when you search that, and then you go down and look at some of the different monographs. And here's one for internal analgesic, antipyretic, and rheumatism drug products. And here's the one that would cover aspirin, and it details how it has an over-the-counter monograph in human medicine. But still, again, this is not approved, but it tells you how the human products are there on the shelf. And here for aspirin is listed there at the bottom. So aspirin and buffered aspirin products. And it states there that they have to be labeled according to the doses for the different buffered formulations that are in another section of the document. We won't get into that. But within this document, it requires multiple things, such as manufacturer and purity, and then how the actual doses that would be on there, because they're generally regarded as safe and effective for that application. Again, those are not approved products by the FDA, so they still don't slip under AMDUCA, but just explains why they're there on the counter when you go walk into your drugstore or your pharmacy. So when we say we should look first at drugs that are labeled for that application in food animals, what do we have labeled for pain and or analgesia in food animals? So I'd first like to thank Dr. Mike Kleinhans for allowing me to use some of his slides. Dr. Kleinhans, along with Dr. Katsia, have done extensive research on NSAIDs and their applications in food animals. And if anyone's interested, I could sure head you towards some of their publications in that. So flunixin comes to us, and we have of course, everyone in here is well familiar with these, but we have the injectable formulation labeled for control of pyrexia, associated with BRD, endotoxemia, and acute mastitis, then control of inflammation and endotoxemia. The transdermal product labeled for control of pyrexia associated with BRD and acute mastitis, and control of pain associated with foot rot. And then we have a combination with an antibiotic, several antibiotics labeled for BRD, pyrexia associated with BRD. So a pharmacokinetic comparison, when you start looking at the need for frequency of administration for pain, this is, again, a table that Mike Kleinhans put together, and you can see the T1/2 values there, the half-life value, the second one, and flunixin IV has about a 3.5-hour T1/2, not that much different from what aspirin has, but the transdermal has a 6-hour T1/2, which would greatly prolong the duration of activity. The Cmax for has 1.2 there, looking at the transdermal product, and your area under the concentration curve is total exposure. And you can see under the AUC that the IV at 2.2 mg per kg, the label dose, gives you an AUC of 13.9, and the area under the concentration curve for the label dose of the transdermal product gives you 10.1. So a little less total exposure, but it comes out in a way that it is flatter and longer, so you have a longer duration of activity. And we'll talk about how that's been measured. But you can see the other products there you're familiar with, the Resflor proprietary product with flunixin included, as well as Hexasol, the oxytetracycline product with flunixin included. So again, the Banamine transdermal product has a label for pain associated with foot rot, so it does have pain on the label for cattle. Has an 8-day meat hold and a 48-hour milk hold. So this is a product that has a label for lactating dairy cattle with an established milk withdrawal time. AMDUCA does say that any extra label use requires you to apply an exaggerated withdrawal time. And extra label use does include label regimen, but for a different indication. So technically, when you go to using it for pyrexia associated with another disease or pain associated with another disease or a procedure, by AMDUCA, an exaggerated withdrawal time should be applied. So how long? If you look here, this is IV versus topical, data from Kleinhans in his dissertation published. You'll see that there's quite the dotted line here for topical. We trade some Cmax, which is our maximum concentration. We trade that quite a little bit, and that is why we have a lower AUC with this, but here we have those concentrations playing out a lot longer there with the transdermal. And if you look at how it comes out, our maximum time for maximum concentration is out over 2 hours, which is a lot longer than the IV, which is instant. Our Cmax is lower, our half-life is about double and longer, and the bioavailability is about 48%. So even with a lower bioavailability, that longer T1/2 still stretches out our duration of activity. And the reason it does that, we'll cover, I guess, next slide. This one is showing what happens when you give it 3 times. and it nicely comes right back up, and you can see it drops, another dose, drops, another dose, drops. So that extends it out quite a ways. Our accumulation factor is about 1.1, which means that at our second dose, we're probably pretty close to steady state, definitely would be by third dose, and we're going to be about 1.1 times the concentrations in those repeated doses as we are in the first ones. But here again, it's showing a 5.2 hour T1/2 in these after the final dose, Tmax 2.8 hours after the final dose, and a Cmax about 1.08. So how long does it actually come out having an effect? This is some MSD data out of Europe, but it's an exudate model where they evaluated ex vivo reduction of prostaglandin E concentrations, and they found that the take home on that was the anti-inflammatory effect was about for 48 hours after a single dose of the transdermal. This looks at the IC80, which is the dose it takes to inhibit 80% of cyclooxygenase activity in an ex vivo model from the concentrations. And that concentration for flunixin in this study was 0.039 micrograms per mil. And you can see here that it's carrying that plasma concentration out here about, close to 36, 40 hours. Next slide is some more work by Dr. Kleinhans showing the prostaglandin E2 suppression. So this was prostaglandin E inhibition in an ex vivo lipopolysaccharide model in 2-month-old calves with flunixin transdermal. And again, their work showed well up there around 30 hours that they were suppressing prostaglandin E2 in an ex vivo test. So all the data suggests that you're good definitely for 24 hours, well out to that day and a half. And again, the data in the model from the European study would suggest 48 hours for that duration of activity. So here we have a drug that's approved has a pain approval in cattle, has a milk throw in dairy cattle, and has been shown to have an effect against prostaglandins and for pain. The other alternative that's been looked at is, again, extra label, and this is a drug that has, meloxicam is a drug that has a human approval. We do not have a food animal approval. We have a human generic, and these tablets display 100% oral bioavailability in cattle, so very, very, very well absorbed. Has a half-life of about 27 hours. So a very, very long duration of activity. The meat withdrawal period in the European Union is 15 days, and Canadian withdrawal is 20 days. Any use of this in a food animal by a veterinarian requires you to work with a source to establish an exaggerated withdrawal time, of course. Typically used at 0.5 to 1 mg per kg orally in feed or as tablets, administered in gel caps. And I say in feed, it would be illegal to put this in the feed of like a herd or a pen. But individual animal, when you're trying to get it in an individual animal, putting it in a bit of feed and let them consume that, I do not consider that being the same as pouring bottles in the feed of a pen of cattle, and that becomes illegal extra label use in the feed. But using feed as an individual animal intake mode, I think would be a minimal issue. So a 30-day ELDU withdrawal time has been suggested by some, and again, for you to have a withdrawal time for that, you should be using a source such as the Food Animal Residue Avoidance Data Bank to establish a withdrawal time based on the labeled data, but this is one that has been used, and Dr. Katsia's work after a mg per kg oral dose suggested a 96-hour milk hold. Again, first time you establish these withdrawal times, don't use this. Go back with Food Animal Residue Avoidance Data Bank and get your withdrawal time information from them if you choose to do this. A lot of people go every other day for a couple days, every day for a couple days, and every other day. And extended use has been done with no detected ill effects. And again, if you look at how this goes, there's some IV and the 1 mg per kg PO. This is down here at 100 hours, staying well above the 1 microgram per mil out there for 100 hours. And when you look at some of the work that's been done, effective analgesic concentrations for 3 to 5 days following a single oral 1 mg per kg dose. And it's been shown to reduce stress associated with cautery dehorning and a mg per kg orally, and reduce BRD subsequent to castration when given at a mg per kg orally prior to the procedure. So those are all out of published papers. I'd be glad to provide you. Amphotericin B-induced lameness in beef calves, 4-day decrease in signs after 0.5 mg per kg PO once daily for 4 days. A lot of work done on it. What we're left with looking at is flunixin is approved for cattle, not only food animals, but for cattle. It has an established milk throw and a slaughter withdrawal time in cattle. It is not approved for the pain for castration or dehorning. It is for foot rot, but it does have a pain approval and for food animals. Meloxicam has quite a little bit of work for it, but it is what's being used is the human generic tablets, so it meets Amduca for that reason. And I will remind you that they're very specific in Amduca that cost is not a valid reason for extra label drug use. One of the things I do consider valid is that it is up to the veterinarian within the course of their professional practice to decide what is and isn't practical for application, and when an application issue would create a problem with the animals being treated and getting the right dose. So that is in the practice of veterinary medicine, and to me is one of the components that tells you whether or not a product will be effective. So that's as good as I know how to lay out the Amduca issues with considering these other drugs. There is another one that we've known of in the past, in phenylbutazone or Bute. Of course, all of those of you who practice on dairies know what would happen if that were to be found on a dairy during an inspection, as that is specifically prohibited from extra label use in dairy cattle over 20 months of age. And also with it, it does have a very extended withdrawal time. Its T1/2 in plasma is about 55, 56 hours. Working with some people in the past, looked at some data to suggest that if you were to check urine for it, probably 40 to 45 days before you would expect the technology at that time to detect it. And of course, the thing to circle back on, another difference between flunixin and meloxicam is flunixin has a tolerance in dairy cattle and in milk. Meloxicam does not. So meloxicam is violative if any is detected, and for flunixin, it is violative only if above the tolerance. And that makes the meloxicam issues subject to how low we can go. And the way we've been going in analytical techniques for the last several decades is every 4 to 5 years, it seems we take a decimal point off what we can detect. So way back in the dark ages, when I did my PhD, we were doing HPLC, UV vis, or fluorescent detection, and we were sniffing stuff out to 50 or maybe 1 microgram per mil. And then we get down to nanograms per mil, and now we're down to decimal places of nanograms per mil, which puts us into the picogram per mil range. And, there's some effort to say, well, what's a practical level that would be there? But, if someone wants to fire up a mass spec and go looking today, very, very, very sensitive. And what's happened is we've developed the technology to pull out our peaks that interfere with the main peak of the drug we're looking at. Just some amazing technology that does us a lot of good, but in cases like this, it detects stuff at very, very, very low concentrations. If you have an interest in the concentrations they're typically testing for, you can access the FDA, FSIS protocol for how their high throughput technique, and you can see their actionability levels on which they consider them positive. And you can see how low they're going on their assays validated for kidney, pretty low. And I would imagine they could get lower in the future. So that's a quick run through. Be happy to have any questions, chat with everybody here. Quick run through on what happened there, why they made that decision, and where we are today. I know there's people with great concerns about that, about the ability to use it. Swine veterinarians have had a lot of conversations about it also. And this letter does make it very clear that the extra label use of an unlabeled drug is not able to be done in food animals, not just cattle. The label, or the letter, was addressing a specific instance, but made it clear that they were applying that to all food animals. Greg, do we have any questions?
Got some questions. Yeah, some, number one is your analogy about driving 63 and a 55 and the highway patrolman said. So this all came about because the FDA realized there was a lot more aspirin being used in dairy cattle because of the high-path avian influenza. Is that right?
That was the trigger. That was the incident that they, caused them to take an internal look. As far as all the internal reasons, I can't, speak to all their internal dialogues, but that was the instigating incident to have them look at it, yes.
And so aspirin's out for us in food animal, and it sounds like from your data that dermal flunixin is probably the way to go in comparison to IV flunixin. Is there any reason why, any indication why somebody would want to go over the labeled dose for dermal flunixin, in your opinion?
I can't speak to how much that would do for efficacy. I don't know if those data are out there. They may be from the company, but I can't quote you right now difference in doses. So you've got a 6-hour T1/2. So if you're going for duration, doubling the dose, if you doubled the dose and instead of a Cmax of around 1 microgram per mil hit 2, that'd add 6 hours to your duration. So in something like that, you're better off with, if you're going for duration, going with the repeated 24 hours later. And Mike's data there, giving it 3 times, shows how that works. But you'd only gain 6 hours on anywhere from Mike's data, the ex vivo data, like 35 hours to the European data, 48 hours, you'd only be adding 6. So I'd go again rather than doubling. And plus doubling, you've got to mess with your withdrawal time too.
And so that comes up to another question we got, is that if you do go extra label for flunixin, and you kept saying you had to find a source to guide the practitioner on withdrawal times, you mentioned FARAD. Mm-hmm. Does FARAD have recommended slaughter and milk withholdings for all the drugs that we use in food animals that we can use as practitioners?
They would have a lot of them. If the data's out there, they've probably got it. I've worked with people there, got to know quite a few of them, and they do a really good job. They've got a good algorithm that figures these things out, and they are experts at finding the data. So if I call FARAD and they don't have the data, I am confident with the resources I have to dig up data that I'm not going to find it, unless it's something I just generated or something like that, and then they'll have it really quick. And one of the things to make clear with FARAD is, they're not there to encourage this. They just help us when we make the decision in the course of a practice.
Are there other sources that a veterinarian could use, or is that what you'd recommend? That's
what I'd recommend. I think they are the they are the pinnacle of that. They've got it figured out and really aggressively go after the data. I know people ask me as a clinical pharmacologist what I'd say for withdrawal time, and my answer is I'd say what FARAD says.
Very good. So another question we had is how do they determine the concern level? We talked about that some products have zero tolerance and some have a tolerance level. How is that determined, and what drives zero versus some levels that are
The
tolerated?
approval process. So if they are approved for a food animal, the approval process, one of the major components of that, of your package, is the withdrawal time. And they go through the process where they have the tox studies they do, and usually 3 species, and they come out with the acceptable daily intake, the ADI. And that's based off, well, the NOEL first, the no observable effect level, and then they come out with, that's on a microgram per kilogram of body weight, and then they put that towards the body weight of a human and come up with the acceptable daily intake. And then they use the the projected intake of muscle, fat, liver, or kidney to come up with what the concentration can be in those of total residues in each one. So for example, they figured we'll eat 300 grams of muscle in a day, or we'd eat 150 grams of liver, or we'd eat so much kidney, or we'd eat so much fat. And then in a dairy cow, those are partitioned because we might eat 300 grams of her muscle and a quart of milk. So the acceptable daily intake has to be partitioned. And then they say which one's going to be our marker residue. In the U.S., we have tolerances. And that's always one molecule. It can be a metabolite or it can be the parent. And then they say how much of that can be there based on the percentage it is, and then they go through the studies to do the withdrawal time. That's the process for an approval. Nothing like that's there for something that isn't approved. So then they just say, we don't know how much you could have per day, so any detected is violated.
Zero.
So that's one of my favorite questions for the students. This is not an approved drug. What's the tolerance? It's the same as the amount of coal imported into the United States in 1461. 0.
You would know that, 1461, right?
Yeah. I don't think they were importing coal in 1461.
Yeah, I don't know. Another question is meloxicam looks like it's labeled for use in food animals in Europe.
Yes.
Is there any work to make that happen in the United States?
I'm not associated with the work. I don't know where it is. I would love to see it. I think all the tools, and when you look at the number of drugs approved for analgesia in food animals in the UK, which is multiple products, and we've got one.
Well, and that's during a time when there's more and more impetus and concentration on animal welfare issues for food animal practitioners. And it just seems strange that we've got now, it looks like we've got one product.
Yeah.
It seems kind of strange that we're expected to be animal welfare stewards, but we don't have the tools to really help us do that.
Well, one of the ways to think about that too is we have one sponsor who's gone to the work of getting a drug approved for pain in food animals. So is the way we reward them for doing that, find every extra label way around that we possibly can. And so in the end, the way the companies look at those is what's going to be our ROI. What's the dignity line? Line for what we need to sell, and that's just the harsh reality of the economics of drugs, right? So everyone who's evaluating the pain med in the United States is saying, okay, will we be able to sell enough to come back with the return on the investment and be able to maintain that? Also, one of the things is the having a model, an agreement on the model that will allow us to say this is efficacious for pain is very important. And that's the first model that was accepted by the FDA for that with flunixin transdermal.
Sean writes in and says South Africa also has a number of registered products. So
yeah. a lot of other countries have a lot of products. Meloxicam is labeled up in Canada, food ants.
I think that's all the questions we have. Anybody out there that thinks of a question down the road, I'm sure Dr. Apley would be more than happy to get an email from you or try to catch him on his phone. And we really appreciate you being with us, and keep looking out for advertisements. We should have another one in March, another webinar. Not sure the topic yet, but again, fill out those surveys and give us some ideas of some topics you'd like for us to present to you. So appreciate it. Have a good day.